Hello students, today's topic is metabolism of basic amino acids. There are three basic amino acids: lysine, arginine and histidine. First, lysine. The characteristics of lysine are it is an essential basic amino acid. It is deficient in cereals like rice, and it is predominantly ketogenic, and it does not undergo transamination. So, fates of lysine. First, it forms acetoacetate. First, lysine is converted into saccharopin, alpha ketoglutarate is required, then NADPH, then saccharopin is converted into L-alpha, amino, adipate, delta semialdehyde, glutamate is released, NADP plus water. So these two reactions is catalyzed by the enzyme amino adipate semialdehyde synthase enzyme, which is a bifunctional enzyme, and its amino terminal domain contains the enzymatic activity of alpha ketoglutarate reductase, and the carboxy terminal domain has the enzyme activity of saccharopin dehydrogenase. So after formation of L-alpha amino adipate delta semialdehyde, it is converted into L-alpha amino adipate by the enzyme dehydrogenase, NADH is required. Next, it is converted into alpha ketoadipate by transamination by the transaminase enzyme. Then alpha ketoadipate is converted into glutaryl coenzyme A by oxidative decarboxylation. Then glutaryl coenzyme A is converted into crotonyl coenzyme A NAD is required. This is also a decarboxylation reaction, and crotonyl coenzyme A is converted into acetoacetate. So this is how acetoacetate is formed from lysine. Next, an important compound known as carnitine is formed from lysine. Carnitine, the chemical name is also known as beta hydroxy gamma trimethyl ammonium butyrate. So this carnitine carries long chain fatty acids from cytosol to mitochondria for beta oxidation, and carnitine is synthesized from lysine in the liver. So how it is synthesized? First, lysine as a part of a protein is first methylated by acetylmethionine, and it is methylated 3 times by methyl transferase enzyme. Methyl transferase to form epsilon and trimethyl lysine when it is still a part of a protein. So it is methylated 3 times. Then the enzyme protease will detach this epsilon and trimethyl lysine from this protein, and the remaining peptide is released to form epsilon and trimethyl lysine in free form. Then it is acted upon by the enzyme hydroxylase, where vitamin C and alpha ketoglutarate are required as cofactor to form epsilon and trimethyl beta hydroxylysine. Then this is acted upon by the enzyme aldolase. Glycine is released and it forms gamma beta-rho-betaine aldehyde. Beta-rho-betaine aldehyde. This gamma beta-rho-betaine aldehyde is converted into gamma butyrobutane by the action of the enzyme dehydrogenase, NAD is required, the coenzyme, and this gamma butyrobutane is converted into carnitine by the action of the enzyme hydroxylase, where vitamin C and alpha ketoglutarate are required as cofactors. So that is how carnitine is synthesized from lysine. Next is formation of hydroxylysine as a part of post-translational modification. Formation of hydroxylysine. So as a part of post-translational modification of the collagen, hydroxylysine is formed. So lysine, when it is a part of tropocollagen molecules, after synthesis, this lysine of this tropocollagen is acted upon by the enzyme lysyl oxidase, lysine hydroxylase enzyme, coupled with conversion of alpha ketoglutarate to succinate, here iron and vitamin C are required as cofactors. So after hydroxylation it forms 5-hydroxylysine. Then formation of L-lysine. L-lysine is formed from lysine. It is also a post-translational modification. So in the tropoplasm molecule, the lysine amino acid is acted upon by lysyl oxidase. Lysyl oxidase, copper is required. There is deamination to form L-lysine. This is the aldehyde form. Now this L-lysine, hydroxylysine and histidine, they together form the cross-linking of collagen. Next is lysine is found in histones. Next, by bacterial putrefaction of lysine, it produces the compound cadaverine. So this is about metabolism of lysine. Next is arginine. Arginine is an essential or semi-essential amino acid, and it is a glucosonic amino acid. It is synthesized from ornithine by the urea cycle enzymes. Fates are: number 1, it forms urea. So when arginine is acted upon by the enzyme arginase, it forms urea and ornithine. So that is how urea is formed. Next is formation of glucose. Now glucose is first ornithine is acted upon by transaminase enzyme. So there is transamination of ornithine. It forms glutamate semialdehyde. Then glutamate semialdehyde is acted upon by glutamate semialdehyde dehydrogenase to form glutamate. NAD is required as the coenzyme. Then glutamate by transamination or oxidative deamination forms alpha ketoglutarate, which enters the TCA cycle, and it can form glucose. This is how glucose is formed. Next is formation of creatine phosphate and creatinine, which have already been discussed in glycine metabolism. Number 4, synthesis of nitric oxide, which is a potent vasodilator, and sublingual nitrate is also used as treatment in the angina pectoris. So how nitric oxide is synthesized? Arginine is acted upon by the enzyme nitric oxide synthase, which contains heme, FAD, FMN and tetrahydrobiopterin. NADPH is required as coenzyme. It forms nitric oxide and citrulline. So this is about metabolism of arginine. Next is histidine. Histidine is also a semi-essential amino acid and it is a glucosenic amino acid. Now the fates of histidine. Number 1 is glucose. So first, histidine is acted upon by the enzyme histidase. There is deamination to form urocanate or urocanic acid. Urocanate is acted upon by the enzyme urocanase. NAD is required as coenzyme. Then it forms 4-imidazolone, 5-propionate, then hydrolase enzyme acts on this to form and form amino glutamate or FIGLU. FIGLU is then converted into glutamate and its form amino group is taken up by tetrahydrofolic acid to form N5 form amino tetrahydrofolic acid, and the enzyme is glutamate form amino transferase. Then glutamate is converted into alpha-ketoglutarate by deamination or transamination, which enters the TCA cycle, enters the gluconeogenesis pathway to form glucose. So this is how glucose is formed. Now assessment of folic acid deficiency is done by FIGLU excretion test. So when there is deficiency of folic acid, tetrahydrofolic acid cannot take up the form amino group. So FIGLU is excreted in urine. It is increased in amount and it is excreted in urine. So this FIGLU excretion test is done by administering 5 gram of histidine 3 times daily at 4 hours intervals. Then after that, urine is collected for 24 hours after the initial dose, and if the value is more than 30 milligram, value of FIGLU is more than 30 milligram, then there is folic acid deficiency. Next is formation of histamine. So first, histidine is acted upon by the enzyme histidine decarboxylase. To form histamine. Histamine undergoes further metabolism to imidazole acetic acid by the enzyme histaminase. Now, this histamine causes vasodilation, smooth muscle contraction, increased gastric acid secretion, and it causes allergic and anaphylactic reactions. It also causes increased capillary permeability. So this is about metabolism of basic amino acids.